All right, ladies and gentlemen, thank you all for being here, and thank you especially to Dr. Shah for being here to speak to you about stem cell-based therapeutics for cancer. I'd like to introduce Dr. Shah to all of you. I know some of you are very familiar with him, those of you taking organic chemistry class. But Dr. Shah is the director at the Molecular Neurotherapy and Imaging Lab at MGH and Harvard Medical School. Dr. Shah is also the director of Stem Cell Therapeutics and Imaging Program in the Center for Translational Research at MGH, principal faculty member of the Harvard Stem Cell Institute in Boston, and associate professor of radiology at the Harvard Medical School. To give you a little bit of his history, over the past 5 years, Dr. Shah has established the first MGH research program to simultaneously integrate therapeutic and diagnostic proteins in stem cells and tumor cells to develop and understand stem cell-based therapies for brain tumors. In recent years, Dr. Shah and his team have published a number of studies in very high-impact journals like Nature Neuroscience, PNAS, and Lancet Oncology, validating the use of therapeutic stem cells alone and in combination with clinically approved drugs for brain tumor therapy. In collaboration with a number of laboratories within the Harvard national and international communities, Dr. Shah's laboratory has recently developed clinically relevant mouse models of brain tumors and stem cell lines expressing multiple therapeutic proteins that can target both cell death and proliferation in both tumor cells and tumor-associated vasculature. The potential for developing novel stem cell-based therapies and imaging markers for cancer in his laboratory has been recognized by many cancer alliances and associations. Based on this, Dr. Shah has received the Young Investigator Award for Alliance for Cancer Gene Therapy, Research Fellow Award from American Cancer Society, and also a number of translational grant awards from American Brain Tumor Association, Goldrich Foundation, James S. McDonald Foundation, and National Institutes for Health of Health. So please help me in welcoming Dr. Shah. Thank you.
Well, this is a pretty elaborate introduction. Thank you so much. You don't look at the what he mentioned. You don't look at it again and again, because then you won't work next day. You'll think, oh, what you have achieved in the past. So you have to put the past behind and look at the next day and do more. And then, when you're in your late 70s, you might want to look at all of it together, because you don't have much to do. Anyway, so it's a pleasure to be here, and it's always rejuvenating to be with young students who want to do something in life. And I think this is one of the studies you might come across, or one of the topics you might see in your lifetime. And the reason this is interesting for us is that this is a fairly new approach to kill tumor cells. Now, what I'll try to do is to simplify it as much as I can. So I really want to thank all these people who are currently in my lab. Actually, not these three, they just left and have their own labs. But currently, these are the people who are in the lab, and without these people, whatever I'm going to show you would not have been possible. So let's focus on. I'm going to make a nice story out of it, and sometimes I tend to go very scientific and not come to the. So I want to make it understand in a very layman's terms, and stop me wherever it's not very understandable. Don't stop me too many times, because it will break the rhythm of the talk as well. So if it's really necessary to stop me, do that. So let's go back to the brain. And I think before we develop any therapeutics, we have to understand what the problem is. And if we're focusing on the brain tumors, if we don't understand the brain tumors very well, it's going to be really hard to develop any therapies. And you will be surprised to know that all the pharmaceutical companies that are developing drugs for cancer, they are never specifically developing drugs for brain tumors. And that's a surprise that we learned like 5-6 years ago, that there's not a single company that develops drugs specifically for brain tumors. So the company that develops drugs for breast tumors and lung tumors, they just get thrown into brain tumor patients. The main reason for why the drugs don't work on brain tumors is because brain is very privileged that things don't go to brain easily. So otherwise, if the bacteria and the viruses start going to the brain, we will not be living that long. So brain is privileged. So if the viruses and the bacteria don't go to brain, that means drugs are difficult to go to the brain as well. So there's a blood-brain barrier between the brain and the blood, and that doesn't allow anything to pass. So that's a good thing, but for brain diseases like brain tumors, it's difficult. That's why the normal market for the drugs, and drug market, is not good for brain tumors. So, but that doesn't mean that we won't do anything for these patients. So let's see how the brain tumors develop. So brain tumors start with so most of the cells in our body are already differentiated. So once we're born, majority of the cells get differentiated into a certain lineage. So you get the neural cells, you get the cells that form the muscles, you get the cells that form the blood vessels, and so they don't divide anymore, except for some blood cell types and some stem cells. So anything dividing in the brain, like the cells that form the tumors, like this is the dividing cells in the brain, and that's how the brain tumors arise. So they become more cells, and then what happens when the tumors develop, they need blood supply, and then the blood vessels come in, and then what happens to the middle of the tumor is the middle of the tumor, because it gets surrounded by so many cells, the middle of the tumor becomes dead necrotic cells, and dead cells in the middle of the tumor are as dangerous as dividing cells because they induce hypoxia. There's no oxygen, and the drugs, if there are any drugs that come to the brain, they don't work. So at the end, what happens is that you get a full-fledged brain tumor. So WHO, World Health Organization, has classified brain tumors as grade 1, grade 2, grade 3, and grade 4. And till grade 1, grade 2, that's very early on, they don't get detected. They get detected only in the later stages. This is the grade 4, a full-fledged brain tumor that has a lot of blood vessels, that has a very high necrotic core. And at that time, what happens to the patient either gets a headache or a seizure and has to go to the MR, and basically then you have 12 to 18 months to live. So, but the other part of the story is that these cells not only form tumors here, but also form tumors elsewhere, because they have the ability to go elsewhere in the brain and form their own tumors. So what is known about all these, not only brain tumors, but also other tumor types, that there are lots of things that are wrong with these cells. So why do these tumors get formed from the normal cells? Because there's a lot of things happening, and I'm not going into the details. There's a lot of things happening inside the cell. There are mutations in the epidermal growth factor pathways and other pathways that happen. But you can use these. For example, there are receptors that are expressed on the surface of these cells that can be used as therapeutic targets, both for. So what we have developed is therapeutic targets only against tumor cells and also the blood vessels. But as I told you, that drugs. So why don't we treat these tumors? Why aren't these tumors getting treated with conventional drugs? One of the problems is that all these drugs do not cross the blood-brain barrier. That's one. And the second is that these drugs have a very short half-life. If they don't reach the tumor quickly, their potency decreases with time. And then they have their own toxicity issues, because they not only, so what you want a drug to do is to kill these cells and leave this normal brain undisturbed. So what is current treatment options for GBMs are that GBMs are the glioblastomas, so that's the grade 4 brain tumor. And these are the current standard of care is, so the patients get operation and then radiation and chemotherapy. And these are some of the therapies that are in clinical trials but haven't gone that far in last few years. So now let me get you into the stem cells and why we can use stem cells to treat brain tumors. So stem cells, again, so you might be confused hearing, oh, stem cells, what are these? There are three different types. The two types are, one is the embryonic stem cells that are primitive undifferentiated cells that come from a 5-day pre-implantation embryo, and then there are the adult stem cells. Adult stem cells are, if you take your bone marrow, you have bone marrow mesenchymal stem cells as adults. You also have neural stem cells in your brain. So those are the adult cells, and it would be great if we can use adult cells because then we don't have ethical issues of aborting embryos. So most of our work is with adult neural stem cells and adult mesenchymal stem cells. And there's the third type of stem cells that you might have heard about that we know from basic biology that once a cell changes from a stem cell to a differentiated cell. So here's the example. You know, when we sow the seed in the soil, it becomes a tree and then a leaf. Do you think a leaf can become a seed? We never thought that a leaf could become a seed again. But in the last 10 years, Yamanaka and others who got a Nobel Prize last year have now figured out that you can take fully differentiated cells, like you can take your skin cell or a leaf, for example, and convert it into a seed. So you can take a skin cell and make it a stem cell. That's called induced pluripotent cells. It's called the iPS cells. So I'm going to spend a few minutes on how we have played with developing iPS cells in the lab, and then I'm going to go back into the stem cell types, adult stem cell types we have used. So here are some of the clinical trials for cancer with adult stem cells in different, so phase 2, phase 3, different phases mean. Phase 1 means very early on, phase 2 means it's progressed to the next level, and phase 3 means it can now become a general medicine for the treatment. So what are the sources? So adult stem cell sources, you can have sources from embryo, as I said, you can get cardiac stem cells, you can get stem cells from the blood, you can get stem cells from the fat, you can get stem cells from pancreas, you can get all these different types of stem cells, and there are now techniques that can isolate stem cells from different organs as well. So it's, if you go to our webpage, it's all detailed there in different publications. So let me talk to you a little bit about induced pluripotent cells. Basically, what you take a fibroblast, a skin cell, any skin cell from a mouse or from a human, and then you introduce factors that can make them stem cells. And in 14 to 15 days, they can become stem cells. So your skin cells can now become stem cells, and we've done this in the culture. So the funny part of it is that they become embryonic stem cells. So from a skin cell, they become embryonic stem cells, and then they become the stem cells. So these cells in between, when they become embryonic stem cells, they also beat. They're like heart cells as well. So they beat in the culture dish. And again, these are some of the pictures that we have modified them with fluorescent proteins. And then finally, from a stem cell, we can make them again neural stem cells. So basically, it's going to be hard so if we want to treat an individual with a brain tumor and we want his own cells, we can't take them from the brain because at 50 years of age, your stem cells are depleted. Now, what you can do, you can take patient's skin cell, convert it into a stem cell, and from stem cell, you can convert it back into a neural stem cell. So then you can use that for the purpose of therapies. So we've taken neural stem cells, for example. So these are not from skin, but these are the adult stem cells, and we have engineered it with fluorescent and bioluminescent proteins. GFP is green fluorescent protein, RLUC is Rhinella luciferase, and you can now see these cells in mice, what's happening with them. So we had two sets of mice, and this is very old study, but an interesting one. So two sets of mice, one that had the tumor and one that didn't have the tumor. So then in both mice, we went with stem cells on the other side of the brain. So brain has two hemispheres, right and left, and on the left side is the tumor, and on the right side we implanted stem cells. And in 2 weeks, what we saw is that the stem cells specifically migrate to tumors in the brain, but don't go anywhere. They only go to the tumors. And in the mice that didn't have tumors, they don't move. So if you do the histology now, take the brain out in these mice. This is the site of implantation where the cells were implanted. This is the migration. So cells migrate across corpus callosum into the tumors, and this is the tumor site. So we've shown this with lots of colors. So this is the tumor. These are the stem cells, and stem cells have a natural ability to home to the tumors because tumors secrete different factors that attract stem cells. Now we've shown this with neural stem cells, we've shown this with mesenchymal stem cells, but the whole process of our lab is to develop therapies for brain tumors. So you want to kill tumor cells with stem cells, but not the normal cells in the brain. So what we do is we take a stem cell, we engineer it with genes that are either anti-proliferative, anti-angiogenic, or proapoptic. That means this stops the proliferation of the cell. This stops the blood vessels to grow, and this kills the stem cells. And then what we want is that these factors that are engineered, these stem cells are engineered with, they are released from the stem cells. They bind to the receptors that are specifically expressed on tumor cells or on endothelial cells, and then they kill them. So one of the genes that we have used is TRAIL, tumor necrosis factor apoptosis inducing ligand. It binds to the receptors on both normal cells and cancer cells, but cancer cells have this intracellular domain that connects it to the rest of the pathway. Normal cells don't. That's why only tumor cells die. So I'm not going deep into the pathway. Now here is the image of the stem cells, the green, and the red are the tumor cells. You will see that the red, nothing happens. So these are not engineered with TRAIL. This is like, you will see that stem cells are migrating to the tumors. So if you wait another 5 seconds, you will see a gradient from the stem cells towards the tumor cells. So that again tells you that in a culture dish, they have a natural ability to find tumor cells. Now, if you take these cells and engineer them to express therapeutic protein like TRAIL, this is what happens. Now, watch the red cells. Within 54 hours, this is a 54-hour time-lapse movie. Within 54 hours, you will see majority of them dying. Follow this cell and this cell, and it will die in 2 seconds. It's waiting for my orders. So basically what is happening is that the green cell is releasing the protein that we have engineered it with, and it's killing the red cells. This is what we expect it to do in humans later on. So if you do the math on this, the green cells are releasing the protein that are killing the red cells, and after day 3, there's nothing left in the TRAIL treated group. In the TRAIL releasing stem cells, most of the cells are dead. Now we've done this in mice. So the model is that the stem cells, the blue are the tumor cells, and you implant the stem cells that are red, and they will home to the tumors, kill the tumor cells, and then any residual cells will be killed again by the stem cells. So we've shown this a number of times that the stem cells releasing TRAIL kill tumor cells specifically. And now, the question always comes, how do you really translate this into clinics? In clinics, what happens when a patient has a brain tumor, 70 to 75% get operated. So this is what happens. So I'll come to that later, but first let me tell you that we have also taken control. So what I just told you is that we have now killed the tumor cells. Now we also want to kill these endothelial cells that are supporting the tumor. And for that we have engineered another protein called thrombospondin 1, expressed it in stem cells, and you see now it can kill the blood vessels, the small blood vessels. And it's like 60% of the blood vessels cells are killed as well. Now, to the story where, let's see how we translate this into clinics. So in clinics, what happens when a brain tumor patient comes to the clinics, 70 to 75% of the patients get operation because you have to remove the tumor, otherwise the pressure of the tumor in the brain will kill the patient. So what happens is majority of the tumor, or depending on the surgeon and depending on where the tumor is, gets removed by the surgeon. And then so we try to mimic this in a mouse model. So green is the tumor, and this is one of our postdocs removing the tumor from the mouse brain. And then, so this is the tumor, this is what is remaining of the tumor, and this is the removed tumor. And if you again do the imaging of this, you see that 60%, 70% of the tumor has been removed. But then the residual tumor that is left here, how do we treat this residual tumor? And we have developed ways now where we can treat this tumor. And I think that's where a lot of our work has caught attention, because you can't put drugs in here. So it's a very simple, let's say, if we have a cavity here, so what will happen to this cavity? It will get filled with blood and cerebrospinal fluid. So anything you put there, any drug you put there, will be ineffective. And people have tried putting drugs there and they have been ineffective. And we tried to put stem cells, being very naive about it. We tried to put stem cells there and they got washed out as well. So what we did is we took gels, biocompatible gels. So we mixed our stem cells releasing trail with biocompatible gels and put them in the cavity. And what these gels do, they cement the stem cells, so stem cells stay intact in the cavity. And now if you do the numbers again, it's stem cells without the gels and with the gels. With gels, they survive for 28 days. And without gels, at day 7, most of them are gone. Now if you do the survival curves and look at the tumor volumes, when mice are treated, the residual tumors in mice are treated, you get almost 100% survival in mice. So this is what we're taking to the clinics now. It's already being under FDA approval. So if this works, so the idea is that when we operate the patient, we will put stem cells releasing trail in the resection cavity with the gel, encapsulated in the gel. And the gel will degrade over time because it's a biodegradable gel, and the trail that will be the therapeutic protein that will be released will be killing these residual red cells. So the other part of the story is, so now we know that there is resistance to different drugs. So every tumor has a resistance to one drug or the other, and there are tumors that are resistant to trail. That's one of our drugs, and there are combinations which can make cells susceptible. So one of the combinations we used is an HDAC inhibitor. So if you look, these cells are resistant to trail, not fully resistant, but almost resistant. And then you add this HDAC inhibitor, and now you see a lot of cells getting killed. So you add trail and HDAC inhibitor, and compared to